Welcome to this instructional video on sodium dodecyl sulfate polyacrylamide gel electrophoresis. You can just call it SDS page. So you've just done some protein purification and you have many samples or fractions that you probably collected from possibly a chromatography experiment. And the problem is, your protein of interest is in probably one of those fractions, but they all look the same because they're all clear. Because most proteins do not have a color. So we have to have a way to visualize or detect proteins in samples. And a very good way to do this is SDS page. So the goal here is to detect proteins, not purify. This is complementary to purification because we allow us to detect which proteins are actually in a particular sample or fraction. Well, this all comes down to pouring or buying a polyacrylamide gel. It is a gel. It is solid-like substance that is porous. It is made from polyacrylamide. And what we do is sandwich this polymer between two plates or two plastic plates, glass or plastic plates, and they all have what we call a lane or lanes. And you load your sample, which is probably, a fraction from a chromatography experiment that has proteins in it. Plus, you're mixing these samples with SDS, and SDS is sodium dodecyl sulfate. It is a detergent, and it does two things. Number one, it denatures proteins or forces them to unfold. So this is going to be a denaturing gel, you may call it. We're going to do this under denaturing conditions. Number two, it gives proteins a large negative charge. And that's going to be very important here because what you're going to do is you're going to load your lanes with your samples, and then you're going to apply an electrical current across the gel, and proteins are going to migrate down the gel. Why do they go down the gel? Because you have, again, a power supply connected to this gel apparatus, and you're going to have the negative end, the negative electrode, and the positive. And the negatively charged proteins are going to migrate toward the positive pole, if you will. Then you run this a certain amount of time, and then you're going to get what we call a banding pattern. And what we know is that smaller proteins migrate further down the gel than larger proteins in a given amount of time. And how do you actually visualize the proteins? Because again, you have to do something to make them visible. There's a staining process, and so typically you stain the gel, the proteins in the gel, with something called Coomassie blue or Coomassie brilliant blue, and to no surprise, it makes the proteins have a blue color because the dye sticks to the proteins and gives them a blue color. So what can we get from this? Well, what we can get from this, ultimately our goal is to identify bands and then this will allow us to calculate the molecular weight of the proteins in a sample. And typically in biochemistry, molecular weight is calculated in Daltons. And really a Dalton is a grams per mole that you've seen in chemistry previously. Sometimes you may see kilodalton. If a protein is very large, you may see it converted to kilodalton. So here we go. How does SDS do the two things that we know that it does? Denatures proteins and gives proteins a large negative charge. Well, let's look at the structure of SDS. It is what we call an amphipathic molecule. It has a polar head group and a nonpolar tail group. And so it is going to be what we call a detergent. Detergents are all amphipathic in nature, and it will, again, denature the protein and lead the proteins to have a large negative charge. So let's take a folded protein. And then proteins fold, and I think we've probably talked about this in class already. Proteins fold in many respects due to wanting to get nonpolar amino acid side chains, think leucine, isoleucine, alanine, away from water. So soluble proteins have a nonpolar core, and that's what I've tried to demonstrate in this kind of sketch here on the left. We have an amino terminal into a protein, a carboxyl terminal, and assume this is a folded protein. And then in the core, away from water, are where we're going to find a lot of the nonpolar amino acids. Well, if you add sodium dodecyl sulfate to a sample of a soluble protein, then you will tend to disrupt the equilibrium away from the folded, and you're going to go to the unfolded state. Let's talk about why that might be. So here's our amino terminal, and I'm just going to draw, a cartoon of an unfolded protein. Why doesn't a protein want to unfold? Because you don't want to expose these nonpolar side chains to water. But what if you have SDS available in high concentrations? Well, that's not too big of a problem, because think about it like this. If you had something like an alanine and a leucine here, and we'll just do two of these, and you have an F and a methionine here, then our SDS molecule, let's abbreviate SDS with a little cartoon with that little squiggly tail. There we go. Polar head group, nonpolar tail. Look at that. The nonpolar tail of the SDS solubilizes the nonpolar tails of some of the side chains that were in the core. So now it's the nonpolar side chains that were in the core are not really exposed to water. They're interacting with the nonpolar side chain of SDS. And oh, by the way, this large negative charge of SDS is interacting with the water molecules that are around. And I could do this for a long time, but I'm not going to. But you get the point, I hope. These little dots are water molecules. So we favored the unfolded protein. And oh, by the way, we know two more things. That we get about one SDS molecule for about every 2 amino acids in the protein. And that once you treat a protein with SDS in a sample, all the proteins in a sample will do this, about 1 SDS molecule for every 2 amino acids, then you're going to end up with a sample of proteins, and they all have similar. Alright, I had to pause for a second, but now we're back. So, if you have a sample of proteins treated with SDS, then we know there's about 1 SDS for every 2 amino acids of the protein. So ultimately what you get are proteins with a similar charge to mass ratio. Because the larger the protein, the more amino acids, the more SDSs that will attach to the protein to unfold it. So this is going to be important because we're going to want to separate proteins not based on the charge, because we're swamping out the native charge of the protein with SDS. We want to separate them just based on size. So if we kind of project ahead a little bit, and if we look over here on the left, let's make sure we understand this. So here we go. We have 3 proteins over here on the left, yellow, green, and red. The yellow is a larger protein and it will not go through the porous gel as well as the as the smaller proteins, the green or the red. Now, all the green, the red, and the blue, where's the blue? I don't know. The yellow, the green, and the red proteins all have similar charge to mass ratios. So they're all going to be moving down toward the positive electrode, but they will be, I guess, through friction, retained, or there's a drag on them. The larger they are, the slower they will move through the gel matrix, if you will. Okay, so we have the larger yellow protein, didn't go very far, and then we have the smaller red proteins that went quite a ways down. Again, we would run the gel a certain amount of time, stain it with Kumasi blue, so they will all look blue, and then we can analyze the gel. So what you typically want to do is one lane of the gel always has to be a standard lane. You can get from Sigma or Biorad, whatever, these standards, and they are a mixture of known proteins of known molecular weight. So we have over here lane 1 is a standard lane. It has myosin all the way down to lysozyme. Well, myosin is 200,000 Daltons or 200 kilodaltons. Lysozyme is 14,400 Daltons. And no surprise, the smallest protein in the whole mixture moved down the gel the furthest distance. But how can we quantitate that? We can calculate what's called retention factor, or R sub F. To do that, you always have to divide the distance traveled down the gel by a constant length. So sometimes we just make the length of the gel, let's call that A. It's just how long the gel is. That's going to be A. And then we measure down from the lanes B, and B was myosin. So myosin would have an RF factor of the distance B divided by the distance A. C is in our unknown. C is something it was in a fraction. We need to know the size of C. Maybe that's the protein that we're looking for. And I think you can kind of tell right away from this that the size of C is going to be somewhere between 31,000 and 45,000 because it banded out between those two bands of the standards. But we need to do this more quantitatively. So RF for C is going to be the distance. The RF for the band in lane, the only band in lane 2 would be distance C divided by, again, distance A, which is a constant distance in this calculation. And you can collect RF values for every band on the gel, and then you will basically do a plot that looks something like this. Okay, you have log molecular weight, so you know the molecular weight of the standards. You know the molecular weight of the standards, and those are the pink dots on this. Okay, those are the pink dots, and you're going to do a regression line. So you know their molecular weight. Take the log of that, plot that versus the RF value you calculated previously. We just talked about that. There should be a linear relationship here. And then you go over, well, you draw a best fit line through that, and then you calculate the RF value for your unknown right here. Okay, you extrapolate up until you hit the line, and then you come on, extrapolate over, and then now you can get the log molecular weight of the unknown protein. So right here, you know this now, log of molecular weight, and then just take the antilog of it. Sorry about that. You take the antilog of it, and you can calculate the molecular weight. Again, SDS page is a powerful method to determine which proteins you may have in a sample. And again, the factor that you are relying on in identifying the proteins is the molecular weight of the protein. And so the banding pattern tells you how pure your sample is. Okay, and then the molecular weights of the bands on the gel gives you the relative sizes of the proteins, and maybe it matches the protein of interest for you. So this is a kind of a broad overview of how SDS page can be used in protein purification. Really, it's detection, not purification.